Okay, very good, excellent. So welcome back, everyone. And I want to echo what Uri said and thank Bill Newsome for giving this talk. It is my firm belief that if we want to understand anything about cognition and especially about action, we really need to look inside the brain and we really need to connect with animal models to do that. So I think I really look forward to this talk. So just in terms of the basic mechanics, we're going to take questions at the end. So I'm going to be monitoring the chat, and then I'm going to try to select some of the best questions. You can always bribe me and send money to my PayPal account if you have any questions that are not very good, but you still want to put them out there. Otherwise, you will raise your hands during the after the talk, and we'll have a lively discussion as well. So in the spirit of enhancing communication between neuroscientists and philosophers, I think it was a great idea to have philosophers introduce neuroscientists and vice versa. So I'd like to invite Adina Roskis here to introduce Bill Newsome.
Great. So we're thrilled to have Bill as our speaker. Bill Newsome is the Harmon Family Provostial Professor of Neurobiology at the Stanford University School of Medicine and the director of the Wu Tsai Neurosciences Institute. He's been an HHMI investigator since 1997. Bill received a BS degree in physics from Stetson University and a Ph.D. in biology from the Caltech, California Institute of Technology. Dr. Newsome is a leading investigator in systems and cognitive neuroscience. He's made fundamental contributions to our understanding of the neural mechanisms underlying visual perception and simple forms of decision-making. Among his honors are the Distinguished Scientific Contribution Award of the American Psychological Association, the Dan David Prize from Tel Aviv University, the Carl Spencer Lashley Award of the American Philosophical Society, and a Guggenheim Fellowship. My personal favorite for its name alone is the Golden Brain Award from the Minerva Foundation. He's given a dizzying number of distinguished lectureships, including the Marr Lecture at the University of Cambridge, the Brenda Milner Lecture at McGill University, and most recently, the Distinguished Visiting Scholar Lectures at the Kavli Institute of Brain and Mind at UCSD. He was elected to membership in the National Academy of Sciences in 2000, to the American Philosophical Society in 2011, and to the American Academy of Arts and Sciences in 2017. Bill also co-chaired the NIH Brain Working Group, which was charged with forming a national plan for the coming decade of neuroscience research in the US. Bill's scientific accomplishments are legion. He can be credited with uniting sensory physiology with visual psychophysics with his pioneering work in rhesus monkeys, in which he simultaneously characterized their psychophysical behavior and recorded electrophysiological signals from putative neural substrates, allowing inferences about the mechanistic underpinning of perception. He also demonstrated with electrical microstimulation experiments that columns of direction-sensitive cells in extrastriate visual area, MT, are responsible for perception of motion direction. His studies paved the way for a generation of researchers to analyze the neural substrates of perception in numerous sensory modalities. More pertinent to this group, Bill has also pioneered cellular-level analyses of perceptual decision-making. Decision-making is the key cognitive link between perception and action. In a series of studies of the neural mechanisms underlying motion perception with Mike Shadlin, he explored how sensory evidence about motion in the world is converted into a binary decision. They identified neural signals in the parietal and frontal lobes whose activity was correlated to visual decisions in a manner expected from the drift diffusion models and human psychophysics. And so he related physiology also to computational modeling. Bill extended this kind of work to cellular analyses of value-based decision-making, in which internal models of reward value are built up gradually through experience of the world. He developed methods that allowed him to measure quantitatively the value associated with particular visual icons by a foraging monkey, and then developed a model for the putative internal decision variable that guided the monkey's choices. His group then, in electrophysiological experiments, identified neural signals in the parietal and frontal lobes that correlated well with this hypothesized internal decision variable, and showed that the same parietal circuits that reflected decision-related computations in perceptual choice also reflected decision-related computations in value-based choice. And finally, in collaboration with the Shenoy Lab, he's been central in applying dynamical systems analysis to neural decision mechanisms. Neural systems within the brain exhibit several key properties of dynamical systems, among them massively parallel processing, ubiquitous feedback signals at virtually all levels of the system, and complex dynamical motifs that can be detected in the coordinated activity of simultaneously recorded units. Bill tackled the problem of contextually sensitive decision mechanisms in which decision rules and the flow of sensory information through the circuit must change from one behavioral task to the next. The first paper from this collaboration, which was first authored by Manti in 2013, identified a novel network mechanism for contextually sensitive gating of sensory information within decision-making circuits. This work has inspired many other research groups to pursue neural population studies within this framework of dynamical systems. Bill is revered and admired by his students, not only for his scientific acumen, but for his generosity, his moral grounding, and his fundamental menschiness. His humility extends to his practice of science. In the words of one of his students, it was all about the ideas with Bill, no ego. Bill may not know this, but he and I are related. Bill is the academic father of Mike Shadlin, and I spent a summer working in Mike's lab. And so, though Mike is not my graduate mentor or advisor, I learned a lot from him, and I'd like to also think he learned something from me. So we're more like cousins, and that makes Bill my uncle. So I'm pleased to present Uncle Bill, who will be talking today about detecting covert cognitive states from neural population recordings in prefrontal cortex. So I'll turn it over now to Bill Newsome. Thanks for coming.
Thank you, Adina. I don't think it's been a long time since I've had, if ever, I've had a comprehensive introduction like that. I need to get that from you. Thank you very much. I almost feel like we should stop and see if there are any questions about the introduction before we go on with the talk. But I'm pleased to be with this group. I know that this is a working group. This talk is sponsored by a working group that's working on the neuroscience and the philosophy of volitional decision-making, volitional movement, and free will. And I'm interested in those conversations, so I'm pleased to share some science with you today, which I'll spend most of my time doing. And then if we have time at the end, I'll make a few remarks about it's not really, I don't know if it's philosophy or not. You guys, there are better people out here to make that decision than me, but a few remarks about how to frame these things and how to think about some of the larger meta issues around them. So as Adina said, I'll be talking about, well, I should share my screen first here. And I should warn all of you that I've had complete Zoom crashes a couple of times in recent talks. So if that happens, I will disappear and I'll reboot my computer and I will come back to you. I'm also getting over a cold and I'm C19 negative, I know. So if I start coughing at you, there aren't going to be any C19 viruses coming through the internet. I want to say that this work I'm going to describe to you today is very much a collaboration, as Adina suggested, between my lab and Krishna Shenoy's lab here at Stanford. And virtually all the experiments that were done that I'm going to show you were done by Diogo Peixoto, who is a graduate student and now a postdoc in my lab and is getting ready to start a brand new challenging job at Facebook. And so he's going on to bigger and better things. But there are a host of collaborators who made meaningful contributions here. You can see their names on the screen in front of you. And I'll just call out Krishna Shenoy in particular. In his group, none of this work would have been possible without their help with the real-time decoding algorithms. And then Ruzbe Kiani is also very involved in this work as well. So let's see if I can figure out how to do this. This little graphic is meant to represent a train of action potentials. If we neurophysiologists drop a microelectrode into the brain and record action potentials from some brain cell, we typically see some very irregular stream of action potentials like this. And it can be hard sometimes to know how to interpret them. So if you see a little cluster of action potentials right here, you don't know if that represents some meaningful event in the external world or a movement of a muscle or maybe some internal cognitive event, or whether it's simply stochastic distribution of spikes given a single underlying firing rate. And neurophysiologists, to cope with this problem in the past, typically execute a number of trials. This is an old paper by Muller and Wirtz in 1976. And every horizontal row here represents a trial, and every black dot represents an action potential. And all of these trials are aligned here in time. So this is time on the x-axis, and they're aligned in time to the onset of a visual stimulus. So when you align this and average over a lot of trials, here's the average or accumulation over trials, you can actually see that many of these little bursts of action potentials are related to the onset of the visual stimulus, and therefore we can actually understand and interpret the neural activity meaningfully. In the same paper, Wirtz and Muller recorded from other cells in the superior colliculus for whom these little bursts were related to the actual execution of a saccadic eye movement. So these are the same kinds of trials aligned on the saccadic eye movement in time, and there's a little burst that precedes every one of these saccades that you can see stands out in the PSTH. So this kind of tool, averaging signals over trials, is what has gotten us as far as we've gotten many times today in neurophysiology. But it's obviously a very unsatisfactory tool for the people who are associated with this network, who are really interested in internal cognition and volitional movement, because so many of the important decisions happen in different ways and at different times on different trials, and all the events leading up to an individual decision happen on individual trials. And when you average across trials, you frequently obscure much of this internal cognition that we're interested in. And you know, my example of this is somebody like Garry Kasparov when he was playing chess, who could sit, you know, for hours in a posture like this and be racing ahead in mind travel to consequences of moves, 10, 20 moves down the game and on the chessboard, while barely blinking an eye occasionally and moving his eyes occasionally across the chessboard, but certainly had a much higher rate of cognitive events happening there than any kind of movements that we can measure externally. So to get to this kind of internal cognition, these kinds of internal states, we've got to have something better than single electrode recording and single unit recording and averaging across trials. So this is the challenge here, that decision formation is an inherently variable process, and it's covert. It doesn't have, frequently does not have measurable external correlates that we can monitor as time goes past. So the current approach to this, and many labs are taking this approach now, as the recording technologies have exploded and given us the ability to record from many neurons simultaneously, we will use these so-called Utah arrays, the bed of nail electrode arrays. This is 100 electrodes. You turn it upside down and insert it into the cortex. And then you can record from, you know, 10 to 200 neurons simultaneously. And the attempt here will be to leverage the statistical properties of this population, this neural population, to examine decision formation on single trials. And I'll sort of, you know, I have to truncate this talk because of the time, and also because I'll be trying to teach as we go along, because I know there are some people, a lot of people here who are not neurophysiologists. And so I'll really just talk about two questions today. Can we reliably decode intentions or something about the internal decision state in real time on single trials, okay, without all the averaging? I promise you to get single trials. And then I'll touch briefly on the second question: can we actually detect cognitive events like changes of mind in real time on single trials? And what we'll use here is the random dot stimuli that Adina alluded to in her introduction, that we've used to study motion signals. And I call these sort of vacillating decision states in random dot motion stimuli. So I'll show you a few of these trials here, and they'll be exactly the same thing the monkeys looked at. There's some deterioration in the signal. The videos are kind of jerky because of playing over the internet and over Zoom, but I think it'll be good enough for you to get the idea. So this first one here is should be an easy motion discrimination for all of you. It's jerky, I apologize for that, but I think most of you could see that that was motion toward the left. And if you were a monkey in our laboratory, you'd either move an eye movement to that leftward target, the red one, or you'd move your arm, a hand movement. In the experiments I'll talk today, it was actually a reach movement. If you thought it was to the right, you would reach to the right target. And the arm movements and the monkey's eye movements are completely monitored, you know, from millisecond to millisecond. Our computer always knows where the eyes and the arms are pointed, and we can read out this decision at the end of the trial quite easily. So next I'll show you a stimulus you should be able to see that that's leftward moving. So this is a little bit more noise dots in there and a little bit fewer signal dots. So some of you, that might have been a little bit harder, but for most of you, I think it would be easy to see that that was a leftward direction of motion. You would choose the leftward direction. Now this one is rightward. Some of you may not be able to see that at all. We're simply reducing the proportion of dots that carry the coherent motion signal and increasing the proportion that carry the noise signal. And you can see that we're going to approach psychophysical threshold somewhere. Some of you may have correctly identified that as rightward motion. Some of you may not have been able to tell. And this next one, this is leftward motion. It's really, that's under 10% coherent dots, and this is right at psychophysical threshold for most practiced observers, including monkeys and humans. And then this last one that I'll show you is actually 0%. This is total noise. So the truth is it's a white noise stimulus in the motion domain, and if you say that it's right or left, whatever you say, you're going to be correct because it will contain some motion in that direction. When the monkeys are performing the task, obviously we reward them for a correct choice. In that last case, where the motion is totally random, we simply reward them with a probability of 50% randomly, whichever one they choose. So the monkey will keep working hard at this because the monkey knows he's got a 50-50 chance of getting a reward, even if he's just guessing in the end. And the animals learn to perform this task very well. And they can do just as well as human observers do, a well-trained animal, under most conditions. And I'll outline the task here for you. And I apologize for these graphics. I usually present this in Keynote, but I transferred it to PowerPoint today in order to add some slides at the end. And these graphics deteriorated for some reason between Keynote and PowerPoint, and I apologize for that. But I think you can see the deal here. This is just the behavioral task. It's a schematic of the behavioral task that the monkeys are performing here. So they're sitting in a primate chair, they're looking at a video monitor, a fixation point comes on right there where my mouse is, and the monkey has to direct his eyes toward that fixation point and touch that fixation point with a finger. Then these two operant targets appear, 2 points of light, and they're on for about half a second, and then the visual stimulus appears, and it can be anywhere from 500 milliseconds in duration up to 1.2 seconds in duration. And at the end of the visual stimulus, when it's terminated, there are two types of trials. In one type of trial, there's actually a delay period that's inserted here after the termination of the visual stimulus, but before the monkey is allowed to move his arm to indicate his decision. There's a random duration delay period here between 400 and 900 milliseconds in duration. And then you get the go signal. The monkey has the go signal. And when he has the go signal, which is just disappearance of the fixation point, then he reaches to the right target or the left target to indicate the outcome of the decision. Now that's 30% of the trials, and I'm really not going to have anything to say about those today. You can ask me about them later if you want to, but most of the trials are going to be this 70% right here, where after the visual stimulus appears, the visual stimulus disappears and the go cue appears simultaneously. So there's no delay period here. As soon as the visual stimulus disappears, the monkey gets the go cue to make the reach. And if he reaches in the correct direction that corresponds to the direction of motion in the stimulus, gets a reward. So that's the behavioral task. Normally in person, I would stop here and see if anyone has any questions, because it's really important to understand what the behavioral task is, and I'll outline the big questions that are going to be coming up in a moment. But I've taken some time with this precisely so that I hope anyone listening is able to understand that. The monkeys, as I said, do very well. This is two different monkeys. This is just behavioral data. It shows the probability correct, the proportion correct as a function of the motion strength. We're down here, 0 is the totally random motion strength, and 80 up here is a very strong, easy to discriminate motion signal. You can see that the monkeys, both of these animals, are operating at near 100% correct for strong motion signals. And then they have these smoothly deteriorating psychometric functions and move down to chance performance close to 0% coherence. So this is just a document for you that everything's kosher here on the behavioral time scale. And as I said, we do simultaneous multi-electrode recordings. We implant 2 of these arrays typically. This is intrasurgical. This is the motor cortex right here. This is the arcuate sulcus up here, for those of you who know monkey cortical anatomy. This is the primary motor area. So one of these arrays goes into M1. The other one goes into dorsal premotor cortex. The data that I'll show you are trying to decode the animal's upcoming decisions on individual trials in real time using signals from both of these arrays. If any of you want to, you can ask me about differences between M1 and PMD at the end of the talk, but I'm going to be considering them as a unit today. And as I suggested, the data comprise units that we can get recordings from about typically 100 units in PMD between these two arrays in PMD and M1. And one recording session, a daily recording session, will get 1,000 to 1,500 trials, and that's one data set. Now, this plot here is a plot of the data that we acquire on a single trial, okay? It looks deceptively like the raster plots that I showed you for the average trials from Moeller and Wirtz, but this is emphatically data obtained on a single trial. And what's plotted here on the y-axis is the units that we recorded from in this experiment. From unit number 1, they're arbitrarily labeled according to the electrode, up to about 220, 210, 220 units that were recorded in this experiment. And then each black dot, as before, is an action potential. So what you can see is every action potential that we obtained from 220 units, starting at time 0, which is where the random dot's onset was, and going out several hundred milliseconds in time. So these are all data that are obtained on a single trial. And the way that we quantify these data so that we can start to understand how we can decode decisions from these data are as follows. So we define a 50 millisecond wide time window. So this goes from 0 to 50 milliseconds. It includes all 220 units. And we simply count the number of spikes that occurred in that 50 millisecond time window for each of the 220 units. And that forms a vector of spike counts. So it's a single vector that has 220 entries in it, which is just spike count from each of the 220 units. And so those are the data we obtained in that interval. But then we just take this 50 millisecond time window and we step it forward in time over and over and over again. And we write down that vector of spike rates over and over again to capture all of the data as a function of time that are obtained on this single unit. So on the single trial, sorry, not single unit, making my own mistakes that I don't want you to make, on the single trial. So what we then do are take these data and we train the simplest kind of linear classifier. This is a straight logistic classifier using logistic regression techniques. And what we're trying to do is predict the probability that the monkey will choose target 1, let's say the rightward target, at the end of the trial, given the data that we have recorded in 150 millisecond interval. So our vector here, that's the vector of responses in one of these 50 millisecond intervals. And we're trying to compute the probability of the monkey choosing target 1 divided by the probability of the monkey choosing target 2. This is the likelihood ratio, the very familiar likelihood ratio, just what's the monkey most likely to choose. And the log of that is called the log likelihood. And this is the equation that describes the log likelihood in the logistic classifier. And you can see that there's a constant term out here that basically amounts to a bias term. Does the monkey have an intrinsic bias to choose right versus left? But out here, what we're doing now is trying to use these vectors of data as a function of time. So here's your response vector of all the 220 cells, and that changes over time, right? The spike counts change over time. And for each one of those time bins, the logistic classifier computes a beta coefficient, which says how useful is this particular cell. So for every neuron, R sub I, all 220 of them, there's 220 beta coefficients or weighting coefficients that basically say how much should the classifier weight the activity of each cell in predicting the monkey's upcoming choice. Of course, we know what those choices were at the end of the trial on the training data. So we collect training data at the beginning of the session, we train up these beta coefficients, and then we start trying to predict the monkey's choices in real time on new trials that did not go into training the classifier. I can say a little bit more about that in a moment if you want to. But this is, you can think of these 220 neurons as defining a 220 dimensional neural state space. And to make that concept concrete, I'm going to invite you just to consider a simplified version where we only have 2 neurons, okay? So forget the 220 for just a minute. Let's just talk about 2 neurons. And on each of a number of trials, we have a spike count from neuron 1 in a particular window, and we have a simultaneous spike count of neuron 2 in a particular time window. So in each of these trials, we can plot the spike count of neuron 1 versus neuron 2, and that locates that data point somewhere in this space here. And this has been coded according to the training data for rightward choices, which are the red dots, and for leftward choices, which are the blue dots. And you can see that there's this discriminant line that we can capture with the classifier. And that's what's described by this equation up here, actually. And so there's a discriminant line that actually allows us, if we only knew the responses of neuron 1 and neuron 2 in this cartoon, we could actually pretty well predict the upcoming choice. Now you take that and just escalate it from 2 dimensions up to 220 dimensions. And instead of a discriminant line, you think about a discriminant hyperplane in that higher dimensional space. And that's exactly what we're calculating here. And then we're simply saying, you know, what's the probability that we can sort out the blue dots from the red dots on either side of that hyperplane as we go from time bin to time bin during the trial. Now, for the cognoscenti out there, I'm just going to say that we calculated these beta coefficients for each 50 millisecond time interval. So that's why we have a function of t out here. But we found that these beta coefficients actually stay constant, pretty constant, during individual epochs. So while the monkey's looking at the dots, they're pretty constant. When you get into a delay period, you need to recalculate them. And when you get into a movement period where the monkey's actually moving his arm, they are vastly different. But most of what we're going to talk about in this talk here just concerns the dots period when the monkey's actually viewing the dots and reaching a decision. So I'm no longer going to show you betas as a function of time, even though we've touched that base. So that's just, if you didn't understand the word of that I just said, don't worry about it. That's for the, you know, people like Gabriel out here who know more about this stuff than I'll ever know in my life. Okay, so out of this, we obtain two very useful metrics. So at any given time during the trial, we can calculate what the classifier believes is the most likely upcoming choice at time t on the current trial. Is it right or is it left? And from this, we can get prediction accuracy. We can say, what is the classifier predicting? And then at the end of the trial, we know what the monkey actually did. And we can say, how accurate was the classifier and how does prediction accuracy change over time as the decision is being formed? More to the point, the more of the data that I'll show you, we actually compute something called a decision variable in addition to prediction accuracy. And the decision variable, you can think of it as the distance of the point from the discriminant hyperplane, and it reflects the model's confidence about the prediction of the upcoming saccade, or in our case, hand movements here, or reaching movements. So for example, on both of these 2 trials, the classifier is predicting that monkey's going to choose leftward, but the classifier is much more confident of that on this trial than on this trial. So the distance from the hyperplane here, which is the decision variable, is going to be larger, and those 2 trials will be distinct from each other. So basically, prediction accuracy is a kind of a digital measurement of how accurately the decision is being predicted, whereas the decision variable gives you some idea about the actual confidence, the strength of the underlying signals and the confidence of the classifier in the upcoming decision. Now, this is the decision variable out here. This is what it reduces to. It's the size of this likelihood ratio. And note that this is in log coordinates. This is natural logs. So if I show you a decision variable later in the talk, which I will do, of 1, that means that that's a natural log unit, that 2.73, whatever a natural log is, that it was that much more likely, the classifier is saying it's that much more likely to choose left than right than left on this given trial. So just remember that it's a log unit, and I'll remind you of that as we go. So what I'm going to show you first are some average data, even though I promised you single trials, and that's where the real gold, that's where the payoff is in this work. But I'm going to show you average data just to touch base with previous literature. And these are trials, it's about 17,000 trials you can see down here in the lower right-hand corner that are accumulated across many, many recording sessions. And what we're plotting here is prediction accuracy. So remember, this is the digital thing. How well did the classifier predict the upcoming decision as a function of time? And this is where the 2 targets come on. The dots aren't even on yet. These are the same trials here, realigned to where the dots come on. And this is the same trials realigned to dots off. Remember, we have to introduce time here because these are variable duration. These trials can be from 400 milliseconds out to 1200 milliseconds duration. So we're realigning things here at dots off. And here we are realigning at the time the monkey actually starts moving his arm. So this is, just sanity check, right? Can our classifier predict these things at all? And yes, it can. The classifier is at chance when the targets are on and there's not been any visual information that's come into the system yet. That's not surprising. It remains at chance until about 150 to 175 milliseconds into the dots. That's kind of the latency for the visual evidence to arrive up there in prefrontal cortex. And then we get this rapid ascent of the prediction accuracy. And by the time you're out to 700 milliseconds, the classifier is operating at better than 90% prediction accuracy. And at the time of dots off, it's essentially 100% accurate. And at the time the movement's going on, it actually saturates at 100% accurate. So there are decisions, the classifier works. And you can predict the monkey's decision several hundred milliseconds in advance of an actual movement of the arm. And this is kind of classic what Mike and I first labeled Mike Shadlin and I first labeled decision-related activity. And so it's highly reliable activity here in premotor cortex that allows the classifier to perform. Now, this is the decision variable. So this is the same data, the same 17,000 roughly trials. And this is that decision variable. Remember, this is the log likelihood that the animal is going to make a rightward versus a leftward choice. Now, we're combining the data, as in the previous slide, we're combining the data here across all the coherence levels. But now we're breaking it down into trials in which the monkey actually made a right choice at the end of the trial versus actually made a left choice at the end of the trial. And what you can see here is that we get a nice separation of these, the decision variable for rightward versus leftward choices out here. And this is, of course, what enables prediction accuracy. But you can get an idea here now that we're talking about, on average, a couple of log units of signal here. More likely, the thing's predicting more likely to choose rightward than leftward. And down here, it goes out to 2 or 3 log units. I'm not sure of the reason for the asymmetry. It's not really important. Here, we've broken things down by coherences. So the high coherence, the easy stimuli, right? This is strong rightward motion. This is strong leftward motion, the solid dark color. And this is another telltale sign of decision-related signals that Mike Shadlin and I pointed out in our early papers on this topic, that as the coherence deteriorates and the discrimination becomes more difficult, the size of these signals and their rate of accumulation falls. So the curves become shallower, the latencies become a bit longer. And this titration here means that the circuits in premotor cortex don't just reflect an upcoming movement, and they don't just reflect the outcome of the decision, but they reflect something about the strength of the visual evidence that gave rise to those decisions. And Mike and his group in particular have done just gorgeous work over the past 2 decades building actual quantitative models, drift diffusion and accumulator models, and showing that you can account for all of this activity and a lot of the behavior besides beautifully with these kinds of models. That's beyond the scope of what we're trying to do in this particular talk, but read Mike Shadlin's papers for that. So that is all touching base with prior literatures to show you that in motor cortex and premotor cortex, we can see exactly the same kind of decision-related activity that has been well documented in the visual motor system previously in LIP in the beginning, but in many places now in the superior colliculus, the frontal eye fields. Josh Gold and his group at Penn demonstrated these signals down in the basal ganglia. They're in the thalamus. So it's a distributed network of areas that are at work during this kind of decision making. Now what I'm showing you, this is where the payoff comes for those of you who've been patient enough to follow this far. And what we're plotting now is this decision variable. And remember, 0 is where the model, the classifier considers rightward and leftward decisions to be equally likely. That's a likelihood ratio of 1. The log of 1 is 0. So there's your decision variable. this is a single trial, and remember these are log units now. So the decision variable is flat up to around, in this case, in this particular trial, about 300 milliseconds in, and then it starts rising steeply, and it actually gets up here towards, fairly rapidly, towards 6 log units. Which is, a remarkably high ratio that the classifier is saying this is going to be a rightward choice. Now, the classifier weakens in its conviction, right? I mean, this is still a pretty strong conviction here, 4 log units, it's going to be a rightward choice. But one of the first things you notice here are these vacillations in the decision variable, which if you think back to those random dot movies, and you think about weak motion signals in particular, many of you will have had a vacillating decision state internally. So when those strong motion displays come on, the high coherence motion, you know immediately that's rightward motion or that's leftward motion. But as that signal gets weaker, you can sense yourself start to be uncertain. And maybe even after you've done enough of these trials, you realize subjectively you're leaning rightward at the beginning of the trial, but there was a little bit more information at the end of the trial, that was leftward. And sometimes you can even change your mind during a delay period as though you're kind of mentally replaying the thing. So subjectively, we know that we have vacillating decision states here, and this is the start of a hint that there may be vacillating neural states underlying this that we can detect not on single trial, not with single units, but by leveraging the population of units to build these kinds of classifiers that maybe we can get in touch with these underlying decision dynamics at the neural level. So if we carry this thing on out to the end of the trial, here's the go signal out here, and aligned on the choice. And here's one where a different trial, this is a different single trial, where the decision variable went into negative territory, indicating an increased likelihood of the leftward choice. And I can tell you actually that the monkey actually chose right on this trial at the end of the trial, and he chose left on this trial. And this is very common to see this kind of buildup of these signals, sort of a steady state during after the go period until the choice gets made, and then to see these strong vacillations during the interim. And a key question for us is, do these vacillations, which remember have no behavioral readout, do these vacillations really reflect the internal decision state of the animal? Are we actually watching the dynamics of decision formation here? Or is this simply noise that's due to the fact that we have 200 neurons instead of 2 million, or due to the fact that we're not recording the right neurons, or that our spike sorting is weird, or any of a number of technical problems that could lead to noise in your measurements? And that is the first question that we really want to address here. Now, sometimes on a small minority of the trials, about 3 to 5% of the trials, these vacillations actually become flips in the signal. So here's a trial where the DV went into negative territory, meaning upcoming leftward decision. But out here, just before the go signal was presented, you actually see this big shift back into positive territory, indicating an upcoming rightward decision. And it stays that way until the monkey makes the movement. And the monkey actually makes a rightward choice on this trial. And in cases like this, where you're actually seeing sign flips and the classifier is going from left to right or vice versa, and here's one that's going from right to left, where it starts out in positive territory and then flips to negative territory, and sure enough, the monkey decides left out here at the end of this trial. These look like, potential changes of mind. Maybe the monkey actually changed his mind during that. And, that's a really interesting thing, a covert cognitive process like a change of mind. And we have candidates here at the neural level. But the question is, how the hell would we know whether the monkey changed his mind in the middle of a trial like that or not in real time? How are we going to measure that? And our basic strategy here, what we decided to do, this is when Diogo and I teamed up with the Shenoy lab, because the Shenoy lab had built these real-time classifiers for their neural prosthetics work. So in that kind of work, you're decoding what a paralyzed person or a monkey with his arm physically bound to his side so he can't move the arm, and you're actually trying to decode which way a paralyzed person wants to move her or his arm. And so they developed all this rapid electronics to get the signals, and rapid software to get the signals off the array and process them and do the real-time calculation of the decision variable like this, and then use that to guide a robotic arm movement in the prosthetic experiments. But what we want to do now is to calculate this DV in real time, which we're doing with on the order of 10 to 15 millisecond lag. And what we want to do is take this and use it to feed back onto the presentation of the visual stimulus. So imagine, for example, here, where this classifier is predicting an upcoming rightward movement, and we don't know if this is noise or real. What if we could stop the trial right there when the DV is at about positive 3.2, and we just stop the trial right there, and we asked, ask the monkey, which way are you going, monkey, right or left, and you're going to get rewarded. So we can stop it there, or we could have let this trial move on out here and stop it when it's predicting leftward, and we can stop a trial. We can stop trials anywhere we want according to the DV that we're measuring, and that's exactly what we did. And so we tried to measure the behavioral significance of these DV fluctuations by stopping the trial that can control DV levels that we read out in real time. So here's the experiment in schematic. There's 2 Utah arrays. We're measuring signals. I've already told you the spikes are counted in 50 millisecond windows. There's some normalization that goes on. Then they're multiplied by those beta weights. So that's the decoder. So we just have the spikes. I'm pointing with my finger at my screen as though you can see my finger. That's a failure mode of Zoom, right? So here it is. This is the decoder. That's the beta weights, right? And we take the data at any given point. We multiply it times the decoder. We just convolve it with the decoder. And that gives us our real-time decision variable. And then we use that real-time decision variable to stop the trial at our desired location. And we say, how well does any given decision variable value predict the upcoming choice? So that leads to this experiment about the reliability. How does the prediction accuracy vary with DV magnitude? And so this is the experiment on single trials. We're going to stop the stimulus presentation when a given value of the DV is reached. We call this the bound. That's a little confusing because it gets mixed up with accumulator models. It really should just be called a trigger level. It's where we are going to stop the stimulus. This is under our control. And what Diogo did, was to define some bounds, and he used 3 to 5 different absolute values per day. And there's a little bit of tolerance around the bound, plus or minus 0.25 dV. And there's a blackout period, an initial 250 millisecond blackout period, because we don't want to take random fluctuations in that first 250 milliseconds. When we know that the information hasn't even hit the prefrontal cortex yet, we don't want to be making predictions on the basis of that. But these are cartoons where in this trial, the DV hit the bound and the trial was terminated right when it hit the bound. Here's one where the DV never hit the bound and it just went out to the predetermined trial length and stopped. Here's another one where it never hit the bound, went out to a predetermined trial length and stopped. On this one, we would have predicted a rightward choice because the DV is above 0. Here we would have predicted a leftward choice because the DV is below 0. And here's one where the DV actually made it all the way to the negative bound, meaning we stopped the trial right at that value. And then Diogo just swept 3 to 5 different absolute values of this per day. And we can show you some actual data here. This is not a cartoon now, this is a real trial. This was the 250 millisecond blackout period. And this is a real trial where the monkey made, a rightward choice at the end of the trial. And on this one, the DV hovered near 0 for the first 200 or so milliseconds, and then promptly marched up to that upward bound and hit this looks like this, well, it was a threshold of +3. So this is a threshold of 3. It hit +3 and we stopped it and the monkey made a choice. If he got it correct, he got rewarded. If he didn't get rewarded, okay? So this is exactly the kind of thing you would expect. Here's a sample trial with a leftward choice where you see the actual inverse phenomena where the DV actually marched into negative territory, hit the bound, we stopped the trial right there. Here's a whole slew of them. So this is a bunch of trials that ended in rightward choices that were terminated at this +3 bound. And remember, that's +3 log units more likely to make a rightward choice according to the classifier. And here's a slew of them where it hit the negative boundary and predicted leftward choice. And indeed, the monkey made leftward choice. Now, your minds are critical and you want to know how much is this cherry picked, right? Did he just show us the trials that look nice with the classifier? Is he showing us all the trials? What's he showing us? And in the next slide, I'm going to show you data from all the trials that were obtained at plus or minus 3 dV bound, but I'm going to show you plus minus 1, plus minus 2, plus minus 4, plus minus 5 as well. I'm going to show you how well we predict accuracy, for example, in monkey H. Now, this is the dV of 3. So dV, the terminating dV, is plotted on this axis, and the prediction accuracy of the classifier is plotted on this axis. And this is taken over the last few hundred milliseconds of the visual period. And when we stop, sorry, it's taken over the last 50 milliseconds. So the classifier is classifying the likelihood of a rightward versus leftward stimulus over the most recent 50 milliseconds before it hits that bound. And what you can see here is for a dV of 3, so these are the data that were illustrated in the previous slide, the classifier is classifying at about 97% correct. So those trials that I showed you in the previous trial are not cherry-picked. For bounds, dV bounds set at plus and minus 3, I mean, the classifier is right on the money here. And what's really interesting is that the classifiers has sustained high performance from a dV of 2 all the way out to a dV of 5. It starts deteriorating when you go below 2, but even for a dV of 1, the classifier is operating here at about 75% correct. So it's no longer 100% correct, but that's an impressive percent correct when you're just talking about, you know, 1 log unit on this classifier. And given that it's predicting these on single trials, remember, I mean, this is averaged over lots of single trials, but the predictions are coming on single trials, and they're coming on only the previous 50 milliseconds of data where that bound was reached. So the prediction, what we would actually expect from the logistic model is this. And you can see that the trained model is hewing very, very closely to what we would have predicted. Now, you can have one of two reactions to this. You can say, well, you trained a logistic classifier and it's kicking out, you know, distributions of choices according to the logistic model. If you didn't do that, you wouldn't have anything here, and you'd be totally right. In one way, this is a sanity check. But I think the fact that it's built on only the most recent 50 milliseconds of data, these predictions, and that these beta coefficients are stable across days, and I haven't shown you the data for that, really suggests that we're onto something very insightful here about the dynamics of the decision formation. And to take that a little bit to more of an extreme, I mean, Diogo tested down to dV of plus or minus 0.5, you know, a half a log unit. And you're still getting meaningful decision signals. You're still getting meaningful readout of the decision down here at about 60% correct. So what that really means, what I want you to take away from this is that when we see fluctuations like this in a long trial that are fluctuating over 6 log units like this, we have got to take those seriously. Those fluctuations really reflect the internal decision state of the animal, okay? Because remember, when we would terminate something at a bound of 3 or 4 or 5 or 6, if we had terminated this, the classifier is going to predict those choices 100% correct, essentially. And even down in this area, these are still meaningful fluctuations down here. There's a lot more noise down here when you get down to decision variables of 1, 0.5, something like that. But these large fluctuations here are very meaningful reflections of the animal's internal process. And that is the key intellectual move of this work and this talk, is to document that we can bring previously covert aspects of decision formation measured on single trials and with single unit recording technology, neural population recording technology. We can measure these things accurately and bring them into the light of scientific inspection. It opens up a whole world of interesting experiments to do, like this one. So this is the second question I told you I would touch on briefly. We're about these putative changes of mind. You know, this small number of trials, 3 to 4% of the trials, where the dV doesn't just fluctuate on one side of 0. I'm waving my hands in the air, showing you fluctuations. You have to keep reminding myself you can't see me. So I can wave my mouse, I suppose. You know, sometimes these fluctuations don't just stay on one side of that dV0 line. They actually cross to the other, so that the classifier mid-trial starts predicting a different outcome. And these are what we call candidate changes of mind. But now we have some tools to go back and actually detect these things in real time and stop the trial. So we did another whole set of experiments where the stimulus presentation did not stop at a given absolute DV value, but the stimulus presentation stopped after we had detected a putative change of mind. So we're decoding these putative changes of mind in real time, and we're trying to detect them. So our algorithm's going along searching, and we're monitoring the fact that on this trial, the algorithm's predicting a leftward choice, leftward choice, leftward choice, but here it switches to predicting a rightward choice. And the way we actually formally define these putative changes of mind, we said we wanted at least 150 milliseconds of stability in favor of one decision, followed by 150 milliseconds of stability in favor of the other decision. So we don't want just single time point fluctuations up and down. We're truly doing some smoothing here to try to get some stability. And then we defined the bounding DV that we would say, okay, we're going to stop here. We've seen a change of mind to our satisfaction. In this case, it's reached a bounding DV of +2. It had reached -2. It had stayed stable for at least 150 milliseconds, shown by these little purple arrows here. And then it had reached +2. And we said, all right, stopping the trial. There we go. So I guess you can hear me when I clap my hands. You just can't see me when I wave them. So this is exactly the experiment that Diogo did. He went back and detected a bunch of these. Here's a change of mind that, putative change of mind that ended in a rightward choice. Here's a putative change of mind that ended in a leftward choice. And this, again, was a reliable finding. Here are some more examples of rightward choices. Here's some more examples of changes of mind that ended in leftward choices. And the next slide will show you, this is exactly the kind of plot you showed a while ago. This is the prediction that we would expect from a logistic classifier. And this is how well the classifier is actually predicting the coming choice following a change of mind. And you can see that it's nice and smoothly titrated. It has the same general shape of the previous data, but it is not on the previous data. In this particular animal, we are not predicting the choice as well after changes of mind. It's still pretty darn good, but not as well as it was with that when a change of mind was not in the picture. And what that means is that the change of mind trials involve something, and it might be the rate of the change of mind. There we have data hinting that that is a possibility, but there's something additional here, that we would have to take account of in order to get up to the levels where we were previously. Now, in the second monkey, we're right on the money. We're right up there at the theoretically expected curve. So there's some difference between these two monkeys in this particular study. But I take this as really encouraging because it means, in both animals, we're actually doing well at predicting following changes of mind. So, we can have more confidence that something really changed here about the neural state. And then you can ask questions about how do the statistics of those neural candidate change of minds compare to statistics of changes of mind that have been documented in human psychophysics. In particular, this nice paper here by Vacillations, Kiani, Wolpert, and Shadlin, published in Nature in 2009, where they had humans looking at the same random dot displays, but putting pressure on a joystick that would move a trajectory, a little cursor on the TV screen up to this target if the human saw a rightward motion, or up to this target if the human saw a leftward motion. And these are actual traces of the movement of the cursor on the screen for the humans on a whole lot of trials. And you can see most of the time the human departs from the origin and goes straight to the rightward target or to the leftward target. But occasionally you get these big arcs, and this has been seen by Matt Kaufman and Krishna Shenoy in monkey arm movements as well. And Vacillations et al. Took these to be changes of mind. And in fact, the human subject said, yeah, I started out toward the left, but I changed my mind. I decided to go right instead. So there's, internal subjective verification here too. And I don't have time to show you data on this, but I'll just point out to you that Vacillations et al. Pointed to 3 statistical regularities in these verified psychophysical changes of mind. And the statistical regularities are: 1, that they're less likely to occur for strong motion stimuli. That's intuitive, right? Well, for those strong stimuli where you know the answer right off the bat, you're unlikely to change your mind. Changes of mind are more likely to correct a mistake. Okay, so you start out with a mistaken choice, and then you correct yourself and get onto the right choice. That is more likely than to start in correct territory, and then as evidence accumulates, make a wrong choice. That's statistically more likely. So also changes of mind are more likely early in the trial when you don't have as much sensory data that has come in. And Vacillations et al. Documented all three of these, and I'll just say in words for you that the neural candidate changes of mind that Diogo and us and our collaborators detected follow these same three regularities and an additional one beside. And this work is under review at a major journal right now, let's say in contention at a major journal right now. And it's up on bioRxiv, and any of you who would like to see it can actually look at this stuff in bioRxiv. So the conclusions that I kind of want to leave you with from the science part of this is that using linear decoding of simultaneously recorded units, we can predict choices with very high accuracy, nearing 100%, well before movement onset. And we can decode this decision variable in real time on single trials, and we can use it to follow internal decision states and to identify candidate changes of mind. And so then in real-time closed loop experiments, the candidate change of mind predicts decision outcome on single trials with an accuracy that varies monotonically with a decoded DV in one monkey just as well as on non-change of mind trials, and the other monkey somewhat less well. The decoded DV, I think this is the big intellectual opportunity here. The decoded DV makes covert transient aspects of the decision process accessible for study. And these are, I would call it baby steps in a good direction, right? We're looking at transient states in a perceptual decision with stochastically incoming visual signal being accumulated over time. And that is not the equivalent of Garry Kasparov playing chess, right? And so there's a lot of ground that we want to get to, but I think this at least shows a roadmap for how to document some of these signals and what they mean. And but in general, I would urge all of us to be aware of the validation problem. It's a very tricky business. So if you have fluctuating signals and you think you have a hypothesis that they might be related to some cognitive process of interest, validating that using behavioral metrics, and that's the only thing we can do with animals. With humans, you can sometimes use subjective reports, but the validation, we should all, be as rigorous and as possible, as good as possible, as high in our standards as possible about accepting this kind of stuff. So that's kind of the end of the science part of the talk. Uri Moaz encouraged me to make some remarks if I wanted to about the main topic of this working group, which is volitional behavior and self-generated behavior. And the general problem that's a conversation that's been going on for 40 or 50 years since Benjamin Libet about free will and the nervous system and the conscious perception of having made a choice. And I don't have things to say specifically about that, but I do have some things to say, and I'll try this out on this audience because it's interesting audience of both philosophers and neuroscientists here. And one of my favorite quotes about free will was this one I pulled off the internet. I'm not a fatalist, but even if I were, what could I do about it? So sometimes that's simpler. So that's the simplest end of the conversation. But Emo Phillips, whoever the hell that was, is where that came from. When I think about freedom and human freedom, what I think matters the most is a sense of self-determination, a sense of autonomy. I think that decisions that we make under duress, somebody has a gun pointed to our head and do it or else, that's not a free choice. There are certain decisions that get made on our behalf that we are implicated in due to the political and cultural structures that we live in. I might not want my tax money to go for certain purposes, and I'm sure we all have our purposes we don't want it to go toward, and yet it goes anyway. And that's not an autonomous decision. It's not consistent with my values, not consistent with my history. And the way that my simple rule of thumb definition of this is that when I talk about self-determination, I'm saying that my behavior is caused, at least in part, and that's an important phrase, at least in part, by my beliefs, my values, my memories, my goals, my aspirations. Now, social scientists take great delight in showing us with very careful experiments that many of our decisions, we think we're making them on one ground, whereas in reality, carefully controlled experiments show that there's a different influence altogether, right? So that's why I say, at least in part, out there. I am well aware that many decisions I make and much of my behavior comes from the fact that I was born to a certain family in a certain part of the country, raised with a certain culture, with certain developmental history to my nervous system, and much of what comes out of me as behavior is never even thought about or codified into values and at least conscious memories, goals, aspirations. But I want to say it's in part. And then a corollary that I believe to be true is that conscious, rational thoughts plays some sort of causal role in my behavior. And we can debate that. We can discuss that. You can ask me about that. I think a key thing here in thinking that our behavior is caused, and caused is a strong word, by beliefs, values, memories, goals, aspirations. To get there and to really understand that, we have to wrap our heads around an idea of non-fundamental causation. And in using that phrase, I'm really following Carl Craver here in his beautiful book published in 2008, Explaining the Brain. And non-fundamental just means causal influences and causal explanations that are above, at some level above the four fundamental laws of physics. So in physics recognizes four kinds of forces, right? There's gravitation, I'm waving my hands again, there's gravitation, there's electricity and magnetism, there's weak nuclear forces and strong nuclear forces. Those are the only forces, to my knowledge, that physics acknowledges in the world. And that is where fundamental causation relies. Now, we all have colleagues who believe that fundamental causation resides at the favorite level of the system they choose to study. So believe me, I have many colleagues in neuroscience at Stanford who are genetic fundamentalists. They think that all of my neurons and circuits and things are fluff, right? Epiphenomenal, and that genetics is where the action, the causal action really is. It's curious how people allocate, where their fundamentalist tendencies lie. But the argument here is that we have to be able to think about non-fundamental causation. And I think the key issue for thinking clearly about that is to think about what counts as a cause, okay? And I'm gonna just stick that in your brains and I'll come back to it. And again, I'll follow Carl Craver in these next few slides, who points out that neuroscientific explanation, and I would say probably all complex system explanation, including all of biology, is intrinsically multi-level. So when we explain some phenomena in neuroscience, which is what we're about in the laboratory, right? Those of us who are neuroscientists, that it's intrinsically multi-level. There is no privileged level. And probably one of the best examples for this is to take the phenomenon of spatial memory, which is something that neuroscientists and psychologists have arguably had the most progress on. And spatial memory is what you use when you're trying to find your car in a crowded parking lot like this up here. I fail this test regularly, by the way, and thank God for little beepers and things that help me find my car in situations like this. To study spatial memory, if that's the thing we want to explain, most of the time in labs today, every time you want to study it, you need to reduce it to some way you can operationalize it and measure it in a laboratory. And in most labs, this is going to be a mouse navigating some kind of maze, whether it's a real maze or a virtual maze, but you got to operationalize this thing in terms of behavior that can actually be measured. Now, one of the great insights of modern neuroscience is that this kind of spatial navigation, particularly in rodents, but increasingly evidence that it may be true in primates as well, is that the structure in the brain called the hippocampus contains spatial maps, and also the entorhinal cortex contains spatial maps, and that damage to the hippocampus can severely damage a mouse's ability to navigate these mazes. So the hippocampus is a neural structure that has a causal role in navigation behavior. But my colleagues who work on synaptic plasticity would say, well, just saying it's the hippocampus kind of begs the question, right? What's really important is how do these synapses change with experience? How synaptically, at a synaptic level, how do things learn? And phenomena like long-term potentiation, long-term depression, habituation, desensitization of synapses. There are many known synaptic plasticity mechanisms now, and they're studied and taken apart at the molecular level. And, you know, those grants start with, it has long been thought that LTP underlies, you know, memory. But, you know, some people say not so fast there, you know, it's not LTP itself. The real secret here lies with a special kind of receptor in the membrane called the NMDA receptor. And it's really the NMDA receptors that act as these coincidence detections that make the neurons that fire together, wire together come true. You know, that's where the real action is. And, you know, my genetic fundamentalist colleagues would say, you know, it's all genes. I put these arrows in here going both ways. And I do that very deliberately, following Carl. It looks like I may have had the date of the book wrong. Maybe it was 2007 here. And in Carl's book, he lays out a strong arguments, compelling in my mind, for multi-level mechanistic explanation in neuroscience. And he defines this thing called mutual manipulability as being core to understanding any mechanism in neuroscience, and I would say any mechanism in biology, really. And this rings true to me as an experimentalist. So mutual manipulability, and he's talking here about components of a mechanism. And he's saying a part, something is a part, a synapse, a receptor, a gene, a circuit. It's a part, it's a component in a mechanism if, okay, this is important, if one can change the behavior of the mechanism as a whole by intervening to change the component, and one can change the behavior of the component by intervening to change the behavior of the mechanism as a whole. And this comes from a line of thought in, I guess, philosophy of biology or maybe philosophy of neuroscience. And Carl's not the first one to be in this. He articulated these things really clearly, in my opinion. But it's called making a difference. This is my colleague Rosa Cow here at Stanford calls these people the mechanists. And I discovered reading this book that at heart, I'm probably a mechanist. And what he's saying here is that something like this, at a biological level, just going in and doing experiments, to be convinced that this is a legitimate mechanism that links these 5 different scales and that they're all happening simultaneously, it's important to be able to manipulate, say, the navigation task. Okay, so pick that mouse up and put him down in a different maze, or change one of the required turns in a maze, and show that the high-level manipulation causes changes in the hippocampus. I mean, if it causes changes in the behavior and doesn't cause changes in the hippocampus, you're probably not looking at a mechanism. Or go into the hippocampus and temporarily inactivate it. And if you do that, and this mouse keeps navigating just fine without the hippocampus, you're not dealing with a mechanism. The mutual manipulability says that the thing is a part of a mechanism if you can manipulate at one level and change the activity of the component, or you can manipulate at the component level and change the activity of the mechanism as a whole. And I think that's why I draw these 2 arrows in here, these double-sided arrows, and that holds all the way down through this hierarchy. So I think that explanation is obviously distributed. I also think that means that causal stories about what makes spatial memory work, causality, relevant causal, what I'd call causal relevance, is distributed throughout all of these levels. So what does this mean? It goes back to my question about autonomy and a meaningful understanding of autonomy being critical for a meaningful understanding of freedom. So I don't think that freedom means that things are uncaused. I believe in causes, right? I'm a mechanist. I believe in causes. I also think that freedom doesn't mean that quantum mechanics is lurking around in the brain, you know, making things indeterminate. I doubt that that's true. So it's the autonomy that I care about, and that I define as self-determination, autonomy, responsibility, and the key issue being what counts as a cause. Now you can see what I meant by that. What I mean when I say what counts as a cause is that in any neuroscientific explanation, it's going to be multi-level. You know that those multi-levels are involved in the same mechanism if you can tweak one and you can see changes in the other. If you don't see that, then you're not on the mechanism. And this is what we do. So this rings true to me, that causal explanation, causally relevant storytelling and understanding of mechanisms runs all up and down those. And this is what I mean when I say if we can find a way to talk meaningfully about non-fundamental causation, and I think we must, then we can take mental causation and responsibility seriously. So these are, these are mental terms. When we talk about that, at least in part, my behavior results from my beliefs, my memories, my aspirations, my values. Those are quintessentially mental and emotional kinds of terms. And I would say that they are high-level states of nervous system, high-level configurations of the nervous system. And it might be, in some cases, possible to reduce those down to the coordinated activity of many, many millions of neurons, but you lose a lot in parsimony when you do that. And ultimately, if they're part of a mechanism, they've got to be mutually manipulable anyway. So I want you to hear something here. Number one, I am not saying that bottom-up causes are unimportant. That would be a stupid thing to say. Explanatory relevance runs both upward and downward, okay? But the point is that both are important. And I actually have an example I'll give. This article in the New York Times startled me back in the 2007 or something like that. The Lasker Award, which is kind of the highest American awards for biological sciences, and many Lasker Award winners go on to win Nobel Prizes. And this story appeared in September of 2006. Psychiatrist is among 5 chosen for a medical award. And this avuncular-looking gentleman here in the bow tie is Aaron Beck, who some of you may know was the inventor of cognitive behavioral therapy. And you have to see why this startled me, because of the history of psychiatry, that psychiatry up until neurotransmitters and neurotransmitter blockers were discovered, and discovered how widespread they were in the nervous system. Psychiatry was mostly a talk therapy-based profession. But when these neurotransmitter systems were discovered, psychiatrists became prescription writing profession. And the talk therapy was largely relegated to psychologists, not to psychiatrists. And here in the midst of that, where I actually had a psychiatry resident in my lab, I can't resist telling you the story. At around in the early 2000s, he was doing a psychiatry residency, but still doing some work in my lab. And he came into the lab one day after being in the clinic all day. And I said, "So what did you learn in your clinical clerkship today? What happened?" And he said, "Bill, I have a new motto: leave no receptor unoccupied." So however many receptors there are in the brain, put a drug on every one of them. And here's Aaron Beck being awarded a Lasker Award for invention of an effective talk therapy. And the efficaciousness of it has been duplicated, replicated many, many times in articles like this, where you have depressed patients, you have blinded evaluation, psychiatric evaluation. A third of them get antidepressant drugs, a third get cognitive behavioral therapy, and a third get both together. And the consistent outcome, not on each individual human, obviously, but on groups of humans, consistent outcome is that cognitive behavioral therapy and antidepressant drugs work individually, but they work better when they're combined. And I think that says something profound about who we are as human animals. I think that, antidepressant drugs are quintessential bottom-up activation, manipulation, turning that knob bottom up. When somebody's writing a prescription, they don't really care if you're satisfied with your job or your relationships, or if you had a bad history with your parents. They just care about getting those serotonin receptors occupied. The person who does cognitive behavioral therapy, on the other hand, doesn't really care about your serotonin receptors. That is a quintessentially high-level intervention. It's a behavioral intervention. It's just as causal, in my opinion, as the molecular level intervention. So these bottom-up interventions modify neurotransmitters and receptors, whereas top-down interventions is explicitly trying to change patients' beliefs about the world and patterns of interacting with the world. In fact, Aaron Beck himself said he called it cognitive restructuring, okay? So beliefs matter, I think. And, so I'm a bit of an unconventional neuroscientist here, right? I mean, neuroscientist tends to be imperialistic these days. Neuroscientist is going to conquer all, and, ultimately we're going to, see where the real causal gears of behavior line. We're going to throw away these folk psychology concepts. Beliefs might be one of them that some people want to throw away, but I'm not that way. I don't think that's true to the biology. I don't think it's true to good clinical practice. As said, if you ignore one of those levels and just focus on the latter, you're doing a disservice to patients, because patients do better with both. Okay, so there's a lot of other things here, but you know, I'll close with this. Understanding the nature, I think, of human freedom is maybe the most important problem facing the neurobehavioral sciences. I think as I get older, Alzheimer's comes more into the picture, right? But human freedom's still up there pretty high. And it's important for obvious reasons of human dignity and social responsibility, but I think also for science itself. And I throw this closing quote out here in kind of a puckish sense. This is one of my two favorite JBS Haldane quotes. He says, "If my mental processes are determined wholly by the motions of atoms in my brain, I have no reason to suppose that my beliefs are true, and hence I have no reason for supposing my brain to be composed of atoms." It's kind of this circular thing when we try to, you know, ideologically reduce everything to atoms. And I think the way we do science is not true to that. And I think my best treatments of patients are not true to that. So the bottom line of this is that I think neuroscientists need to be careful about the way we talk about these issues. Because it turns out some people actually listen to us and think that what we say is true. And we need to be careful about, you know, doing our science and communicating our science, but keeping the whole person in mind. And I'll leave it at that and stop. And I know that I've gone over my allotted time here, and I apologize for that. But there it is. Who takes over now?
Okay, so thank you very much. I am clapping here on behalf of all the attendees. This was quite fascinating. There's a lot of questions here and not a lot of times. I was instructed to invite Hans Lichtenstrom first to ask some questions. So Hans will go first, and then I'm going to invite some of the people who are raising their hands. There's a lot of really very good questions in the chat and the Q&A. I apologize, we probably won't be able to go through them all, but just raise your hands if you actually want to speak. Otherwise, I'll choose some questions from the chat. But first, Hans.
Yes, thank you very much for a very interesting presentation, which appear to be actually two different talks in some sense. Yes. And it would be, you could ask many, I could ask many questions, both from technical to philosophical on issues. But I just wonder, how did you get from the first part to the second part? Because it's not quite clear how you relate what you said about the predictability of choices in these monkeys to your discussion on free will. So I think that is important to make clear.
Yeah, it was a big jump, absolutely. And those are two different talks. You know, there have been some lecture series where I'm invited to give multiple lectures on these topics, and I can actually knit them together a bit better. But Hans, thank you for the question. And just let me say that, you know, if this were knit together better, it would be knit together better if in the second part where I was talking about explanation and free will, if instead of using the spatial navigation example, if I had used a perceptual learning example, right? So that I take the dots work and I say, you know, we want to study perception, we pick a visual stimulus. We know that this area MT is involved in it, and you know, we can stimulate these columns and change the animal's decisions, or we can stimulate the stimulus and change what the MT neurons are doing. And I think if I had used that instead of the spatial navigation, it would be clearer. So the connection between them is that whether I used visual perception or spatial navigation, or if I used auditory localization or whatever I used in that example, the message in the end would still be the same, that if we're looking at a mechanism, that mechanism spans easily 5 or 6 different levels of organization, from the genetic to the molecular to the synaptic to the circuit to the brain structure and on up to behavior. And how we talk about those things. So I can do really beautiful reductionist experiments in the lab, and methodologically I am a reductionist. But when we get over into our understanding of mechanisms and what we pronounce to the newspaper reporters or what we write in the textbooks at the end of the day about what spatial navigation really is or what visual perception really is, you know, that's not everything you think it is. You know, it's really the activity these particular neurons and this particular brain structure, you know, it's that move from methodological reductionism to kind of ideological reductionism that I'm taking issue with. And I think that neuroscientists, I see quotes from people in the newspapers or in books all the time, you know, that the folk explanation for why you do something is X, but we know that the real neural explanation is Y. But, you know, some chemist would come back and say, well, you guys think the neural explanation is Y, but really the molecular explanation is Z. And I object to that entire process. Now, I want to know mechanisms, because the only way you can heal diseases is if you, well, not the only way, the best way to heal diseases is to understand mechanisms. So that is kind of the link. And I apologize if I did not, well, I obviously did not make it clear enough, but the link is, it's going from how I am defining decision-making and studying it reductionistically to how I see those findings in a larger program of mechanistic understanding of the brain and the proper way to talk about causality.
Okay, thanks. But do you say that the change of mind that you see in the monkeys is that a sign of free will in those monkeys?
You know, I would have to unpack, we would have to unpack what you mean by free will, okay? I think that it's not necessarily a sign of free will. I think that you can get pathology that leads to changes of mind, right? If you think about Phineas Gage, the guy who got the railroad pike shot through his head, he could never hold a job. He couldn't even finish a conversation because he, in essence, changed his mind about what he wanted to talk about or where he was. And I don't think that's an example of freedom. I think that's an example of a very, very sad lack of freedom after his nervous system was impaired. So I don't think changes of mind, I would not tout them as free will. I would tout probably perseverance on a task, and perseverance through time is maybe one of the most salient metrics of human freedom. It's a paradox, right? That freedom frequently requires discipline. And in giving up freedom to do some things, in acquiring freedom to do something, you give up freedom to do other things. So there's some paradoxical things. But no, I wouldn't use change of mind as an example.
But still, you would assume that monkeys do have free will, right?
I think to the extent that their behavior is determined by their beliefs, their aspirations, their values, their memories, their goals, then they would have some meaningful degree of freedom. I think humans have much more degree of freedom, right? We have more behavioral possibilities than a monkey does, but some meaningful degree, yes, I think so.
Okay.
I'm sorry, tell that short questions are not my short answers are not my forte. I apologize.
Hans, I apologize. I will have to, I want to switch to, there are many questions. Sure. So if Bill allows, we'll extend the talk a few minutes to try to answer a few more questions, if that's okay with you.
I'm fine. I have no commitments after this.
Okay, very good. Yeah. So let's go, let's start with, I see many hands raised here. So let's go with Liad first, from 9 p.m. in the evening in Israel, probably.
10, almost 10 actually.
10, okay, sorry, go ahead.
Thank you. That was very, very interesting. And I wanted to ask about these changes of mind. Mm-hmm. You said that these fluctuations are meaningful in the sense that if you stop the trial, then you could use them to predict behavior. But do you think they're meaningful also in terms of their source? So how do you explain these changes of mind? Are these caused by random fluctuations that is noise-like, or are these coming from another source, another area that causes the monkey to change its mind? So.
Yeah, so you're asking a really, really great question, and it's deeper and a little bit weirder than you even imagine. I think, you know, one of the things about this particular stimulus that we're using in this particular task is that the stimulus itself really is stochastic. So, you know, if you say, I want 5% coherent dots to the right, and you measure it in 100 millisecond interval, you know, the dots that actually get plotted on the screen, it might be 7% to the right. And then the next was 3% to the right. And, you know, it's a distribution. And it fluctuates from interval to interval. So one notion here is that you're just, you know, you're just literally integrating the visual evidence that comes in. And because the stimulus is stochastic, on some trials, you will actually get net motion in the opposite direction. And the hardware inside the brain, the neurons, the wetware, may be perfectly following the stimulus and still make changes of mind and still make mistakes. Okay, that is true. But that leads to the natural scientific question. Well, you can follow these fluctuations now in the decision variable. You know what dots you plotted on the screen. You kept a record of that during your experiments. How well are these fluctuations in the decision variable correlated with fluctuations in that motion energy? And there's some assumptions that have to go into that calculation, but those assumptions are pretty well worked out in the motion vision literature. And the answer was alarming to me. I mean, there are correlations with the motion energy, but man, it is not nearly as strong as I would have thought as a vision scientist. A lot of those big fluctuations are coming from someplace else. And I don't know where else. And I don't know, like in premotor and motor cortex, they're being bombarded with signals, right? That I have nothing to do with. The monkeys are squirming in the chair and they're doing this and that and the other. Attention is probably coming and going, all kinds of things happening, and I don't know what it is. But I do know, and I showed you the evidence, that those fluctuations are pretty tightly tied to the monkey's internal decision state. So wherever they're coming from, whatever's driving them, they're really having effects on the monkey's decision state. And I can tell you a lot of it is not coming from the visual stimulus itself. So I call it, I say this flippantly in the lab sometimes, and I get scolded for even saying this, but I call it cognitive dark matter. So it's stuff out there that I can see its influences on the ends of my electrodes, but I don't know what it is or where it's coming from. It's important to study for the future.
Thank you.
Very good. Thank you. Adina.
Yeah, thanks so much for the talk.
Thank you for the introduction.
This is my question is a little related to Liad's, and that is that, it seemed to me that what you're showing is a multi-electrode version of the kinds of stuff that Mike and you showed in LIP. You get these same accumulator diffusion to bound models in multiple areas, and I'm wondering what you make of that. I mean, I think intuitively I would have thought there would be a place where the decision was made, but to see it in motor cortex I found really somewhat disturbing, because I thought of motor cortex more as a place where you read stuff out from somewhere else. So are you seeing these accumulators in motor cortex as integrating information from other accumulators and other places, or what's your big picture?
Yeah, Adina, it's a great question and complicated. Number one, yes, this early activity in motor cortex has been seen in many labs. So if you insert a delay period, you tell an animal where he's going to have to reach to, you insert a delay period, and then you give him a go signal. Many labs see this delay period activity in motor cortex. And, there's always been a question ever since Evarts first discovered this stuff in the 1960s about why, if you have this activity here in M1 and PMD, why are the muscles not moving? This is motor cortex we're talking about, for crying out loud. And there's been a gradual appreciation over the last 50 years, more and more, that premotor and motor cortex are deeply reflective of and involved in high-level cognitive planning. And human patients with premotor lesions, for example, have very specific deficits in being able to initiate movements and things like that. So I think that these signals, if you put a gun to my head, I would say I don't believe that the actual accumulation for the decision is happening in motor cortex, but I think that motor cortical activity is reflective of it. It's a window onto things that are happening in a large distributed network. And there may be a purer form of the actual accumulation process someplace else, maybe in an area more tightly linked to sensory areas, possibly in LIP or possibly in basal ganglia. I'm not certain, because one thing we do know is that there are additional signals. Our DV, our decision variable we're reading out of motor cortex, includes cells that come on late in the trial and are not influenced by the coherence of the stimulus. Okay, so that's useful to our logistic model in predicting the classification, so it gets swept into our DV. But those cells that come on really late in the trial and don't reflect coherence, they almost by definition cannot be a part of the accumulation process. So I think there may be earlier areas where a purer form of accumulation exists. I think we're seeing activity that reflects the accumulation process and adds additional motor signals on in besides. But I think the primary function of motor and premotor cortex are to get the system in a state, in the dynamical system sense that you alluded to in your introduction, Adina, for smoothest execution of the upcoming movement. That's the ultimate outcome of the activity in motor cortex. It's not the role that a motor cortex evolved for, to integrate that kind of sensory evidence. It may be one role, but ultimately it's got to get the cord and everything else tuned up to execute movements. There's another experiment that's been done in Mike Shadlin's lab and in Krishna Shenoy's lab that I think is very telling, Adina. And the one from the Shenoy lab is published. I'm not sure whether Shishruth and Mike have published their study yet or not, but they did an experiment that should have been done 20 years ago. They just changed the order of events. They showed the monkey the fixation point, so the monkey fixates. Then they show the random dot pattern, and the monkey has to make a decision about the random dot pattern. And then they turn on the two targets. So they're just flipping the order of the motor targets versus which one comes first, the dots or the two targets. And when they flip that order, and when Megan Wong and Krishna and Chan Chandrasekharan record in premotor and M1, there is none of this activity, or very, very little of this anticipatory decision-related activity in motor cortex if those two reach targets are not up there. My jaw almost hit the floor when I first looked at those data, which, nevertheless, the monkeys are looking at the dots and they're making decisions. And when the motor cortex comes alive is when the two targets come up there, one of which is going to be the target of an eye movement. And, to me, that suggests that motor cortex is, premotor and motor cortex are not the place where, certainly not the place where integrating the evidence and reaching the decision is taking place. It's a place where you can monitor that process, but it's not there. I think Mike and his postdoc, Shishruth, have a different interpretation of that finding, and I'm not sure whether their paper is out yet. But again, you asked a question just like the previous one that is on the money, it's deep, and it's even more complicated than you were aware when you asked it, I think. So complicated answer to a good question.
Thanks.
Very good. Thank you. John.
Hi, Bill. That was great talk. Really interesting. I was just curious because I felt that you call it a decision variable.
Yeah.
By the fact that you call it a decision variable, someone who's not really into the classifier and neuroscience business might get the wrong impression that you are actually at an earlier stage, that you are actually reflecting some kind of decision stage. And while you presented this work, I mean, I'm really fascinated by it, but I was wondering whether if you'd followed the Vacillations idea, that you kind of just have some kind of continuous output of where your leaning is, where you think the tendency is like a graded signal reflecting your current judgment of the probability of it being one or the other direction. Then you could take the joystick position, and you could basically presumably extract a signal that is virtually identical to what you get here out of motor cortex. So I understand that the kind of decision-making literature has often conflated the motor output and the decision stage very heavily, because I mean you have to do the kind of experimental manipulations you just mentioned. I was just curious because you just said it's clear that these signals have an effect on the monkey's decision state. And is that really the case?
If I said that, I shouldn't have, because I don't know whether those signals have an effect on the monkey's decision state. I think probably what I said is they reflect the monkey's decision state.
Okay, so they're the outflow.
Those are very different things. So reflecting it is very much in line with the picture that you drew from Vacillations, that there is a continuous process that involves accumulation of evidence, but also some dark matter that I don't know where the hell those signals are coming from, but they reflect the animal's decision state. But I presented no causal evidence in here at all, right? So I would not, if I said that, I should be.
Okay, thanks.
I should have my knuckles wrapped, which you're doing.
Okay, Aaron.
Hey, thank you. Thanks for a really great talk.
Two talks.
I have two really great talks. That's right. I have just a simple question. I think one of the participants also raised this question, I noticed. Could you use the decision variable to detect when the monkey had made up his mind, in addition to discriminating what choice he was making?
Yeah, that is a really interesting question. There are multiple ways to approach it. Let me tell you about the one that we've done. And I omitted this from the talk just because there wasn't time. And these are experiments. So they're if I have a longer time to give this talk, I have experiment 3 in addition to 1s and 2. There's experiment 3, and this is an experiment that was done by MD Ph.D. student here at Stanford named Jessie Verheyen, and she works in Christian's lab, but very much collaborating between my lab and Christian's lab. And Diogo was also involved in this. What they did was to, it's a variation on experiment that Mike Shadlin, Rusbeh Kiani, Alex Huck have done over the years, where you set a stream of dots in motion. So your stimulus is unfolding, and then you put a little pulse, a little extra pulse of motion into it. So let's say I'm showing 5% rightward motion, and I'll sneak a little pulse of an extra 5%. So it could be 10% to the right, or the pulse could be to the left, so that you have a little time period there where you have, in effect, 0% motion, right? And we try to make these things very subtle. So we have to be able to see it in the behavior, that we shift the psychometric function around. But the question that we asked was, once we became convinced that we could see effects of that pulse, both at the behavioral level and at the neural level, the question then became, when you stop at a particular DV, like 1 or 2 or 3 or 4, when you have higher DVs, you presumably have a greater level of commitment to the decision. Certainly the classifier does, right? And this is something, that John rightly pointed out, that should have been clearer about, that DV is a DV for the classifier. It's not in any way one-on-one the same thing as what's going on in the monkey's brain. But the question that Jessie asked was, when you stop the trial at a particular DV, do you see any difference in the effects of the pulses, those extra pulses of motion energy, right? Because in a straight linear accumulator model, it should not matter what DV you're at. You put in a little extra pulse of motion, it should get integrated. The DV signals at the neural level should change, the behavioral level should change. All you're doing is accumulating evidence, right? But under attractor models, or any kind of models that have nonlinearities in them, potential nonlinearities in them, you might accumulate information up to a certain bound and then make a decision, a preliminary decision, and be resistant to new information that comes after that. In attractor language, you fall into some attractor well, and the new information come in doesn't allow you to emerge from that well with the speed that you went into it. So we were looking for those kinds of nonlinearities, and we found them. We found that the pulses are more effective early in the trial. So as the trial goes on, the system becomes more resistant to the pulses. And also the effect of the pulses changes as a function of the DV. So if you put in the little pulse when the DV is +1, -1, something the monkey's a little uncertain still, you get bigger effects of the pulse than when you put it in for a high DV, when the monkey may be committed. So this all smells like some kind of nonlinearity, whether it's attractor dynamics or whether it's a different kind of nonlinearity, but something that might correspond to the tentative commitment to the decision. So that, in our hands, comes in where we see the pulse fading away with its effect as a function of time. It comes in around 500 milliseconds. Just that data, by the way, are up in that bioRxiv paper, although there's a heavily revised version of that paper currently under revision. So I don't know that I'd really recommend any of you go charging off to the bioRxiv paper, because it changed a lot during revision. It's a good revision process, a good set of reviewers this time. So another way to do that, to do your experiment, would just be to do a reaction time thing. So let the monkey decide whenever he wants to go, and see if the monkey decides consistently, what's the distribution of DVs on trials when the monkey decides to go, and does that point toward an actual decision time? And that would be an interesting experiment. I think we have some reaction time, a modest amount of reaction time data in the tank, but I think that would be another good experiment to do. But you know, this is all murky, right? Exactly how the decision gets triggered, when the commitment gets made. Reaction time probably gives you as good as anything to hang it on, but there may be levels of subtlety even deeper than that. Sorry for the long-winded reply.
It's okay.
And good to see you. Good to see you, Aaron.
Good to see you too. Thanks. Thanks very much.
That was very good. So I feel a bit torn. I think I'm going to allow one more question from Walter here. Walter, if you can unmute yourself. So I apologize. I have to say that there are 15 fascinating questions in the Q&A. There are people raising their hands in the attendees. There are also more questions in the chat. But I don't want to take advantage of Bill's time, and I think he's been very generous to stay with us for this long. So I'm going to take only one last question from Walter, and I apologize to everyone else. Walter.
Gosh, I think I've got 4 questions. Hi, Bill.
Hey, Walter.
I think I've got 4 questions in the chat, and I'll email you about them. But the one I'll ask now, just because it's on my mind, is have you looked at the eye direction or gaze or attention within the dot pattern as an explanation of the change of mind? Because one can imagine that they're focusing on a certain subset of the dots at one point, another subset at another point.
Yeah, that is possible, Walter. I don't, you know, usually in our experience, the fixation point is here. So the monkey's looking here and the dots are up here. So the monkey cannot, in a literal sense, move his eyes to look at different parts of the stem. I'm using my hands. Oh, you can see me now.
Yeah, we can see you now.
Yeah, exactly. You know, so the monkey in our test, if he tries to move his eyes up to the dots, the trial gets aborted and terminated. So his eyes have to stay on the fixation point and his finger has to stay on the fixation point until he's given the go signal. Still, covert attention could be moving around, you know, and I have done tens of thousands of these trials myself as a subject. And I feel like I've tried different strategies. Okay, I'm going to pay most attention to the dots closest to the edge, closest to my fovea, or I've actually tried defocusing my eyes, just make it a blur up there so I can't even see individual dots and see if the motion comes through better that way. But we've never been able to demonstrate any mechanism like that. You know, the best we can do, the monkey has to keep his eyes, he never keeps his eyes exactly on the fixation point. He has a little fixational, you know, a certain amount of latitude that we give him. In some animals, I've seen kind of cheating toward the edge of that box, but I don't think it's attending to a particular region of the stimulus. I think it is moving, it's just trying to bring the whole thing closer to his fovea, closer to his center of gaze, as all primates want to do, right? That's what whole eye movement system is for. And here we are suppressing it in service of, you know, these more abstract questions we want to ask.
Yeah, I think the important philosophical lesson from that is, or the question that it raises is, are these changes of mind based on reasons that they get from the evidence by looking at part of the signal or maybe a certain temporal stage of the signal, in which case they looks like they're in control and they're just responding to the evidence, as opposed to this view that it's just dark matter, this stuff from the outside that's making them go one way or the other without them having any control and without it being rationally guided.
Yeah, this goes back to, you know, one of the earlier questions about the fluctuations, the source of them. And, you know, as a vision scientist, I would love to be able to stand here and tell you that we've measured the visual stimulus, the responses to the visual neurons, and the fluctuations in these decision-related neurons. And it's the same fluctuations in the visual stimulus that are going all the way through the system, right up to these. That would be a beautiful story to tell. And it's just not the right story. It's not the way it is. These fluctuations, as nearly as we can tell, unless there's some better experiment or some better analysis of our data that some other people can come up with, those fluctuations are driven by things that are not in the visual stimulus.
Thank you.
Yeah.
Okay, so I want to thank everyone again, and in particular, I want to thank Bill for 2 beautiful and very insightful talks here. That was quite fascinating, and I particularly appreciated also all the discussions and your philosophical comments as well. That's a side of you that I didn't know, so that was fascinating. I want to thank all the attendees and all the panelists as well for very insightful questions. One more time, I'd like to apologize. There are lots of really very profound and important questions in the Q&A. I'm sorry I didn't get to address, to allow you to ask all of those questions.
Gabriel, maybe one thing we can do is if Bill is willing, we can send those questions on to Bill, and if he has time to answer some of them, then he might be able to do that. That's the only thing I can think about.
Yeah, you can send them. I mean, I have tried copying and pasting from the chat stream before, and it doesn't work.
Yeah, we'll send those to you. But
I don't know how we capture them. You might have to capture them with a screenshot or something. I can try. You know, oddly here, right in the middle of shelter in place still, and approaching the July 4th weekend, I'm desperate for time. Part of it's because I have half-time responsibility for childcare until daycare is open up again. But I'll try. I'll tell you I will try, but don't take it personally if I don't answer. Just
very good. Thank you very much.
Okay.
Thank you very much.
I want to thank everyone one more time, and I look forward to the continued discussions on Friday.
Okay. Thanks.
All right. Thank you very much. That was fantastic. Very good.